Hi, my name is Darya, and today I'm going to be walking you through how I made E. coli survive on streptomycin media using CRISPR-Cas9 technology. So I recently purchased a DIY CRISPR gene editing kit from the Odin, and I really just wanted to show you guys how that works. So this is what the kit comes with: a micropipette, petri dishes, a microcentrifuge tube rack, pipette tips, 2 tubes of agar medium, one including streptomycin and kanamycin, as well as a 50 milliliter bottle for measuring volume. Inoculation loops, microcentrifuge tubes, a transformation buffer, Cas9 plasmid, gRNA plasmid, template DNA, and 2 tubes of non-pathogenic E. coli, as well as sterile water, and gloves, and a glass bottle. I'm first going to start with making my agar plates. This will allow our bacteria to grow and to give it nutrients it needs to thrive. I'm going to label each of my plates with either streptomycin or just lysogeny broth. This will help me in the long run when I do actually edit the genes of the E. coli. Now that I'm done labeling my plates, I'm going to prepare the lysogeny broth by adding it to the glass bottle provided by the kit. Then we're going to add 150 milliliters of tap water into the mixture. As you can see, the mixture becomes a cloudy yellowish color as the LB agar dissolves. I'm then going to shake it to dissolve any clumps, and then we're going to put it into the microwave until the cloudiness is gone and it is a clear but still yellow liquid. During this step, be very careful that the mixture does not start boiling, and take it out of the microwave if it does. As you can see, it's turned into a clearer color, and now I'm going to set it aside and let it cool. Now that the mixture is cooled, I'm going to pour it into my petri dishes. I'm going to make sure that I don't overfill the dishes and only fill them up to about the halfway mark. I'm going to repeat the same steps with my LB Strep Can Arab medium. After these are filled, I'm going to leave them in the fridge overnight, and I'm going to store them upside down so that the condensation does not affect the agar itself. We're first going to take our dried DH5 alpha tube with the E. coli inside. To access the bacteria, we have to add 100 microliters of sterile water to the bacteria tube. This is because our E. coli comes freeze-dried, so we wouldn't be able to use it without adding water. I'm then going to shake the bottle for a minute just to ensure that the E. coli is dissolved in the water. I'm going to repeat this step for another tube of E. coli. I also prepared my bacterial RPSL K43T DNA template, my gRNA plasmid, and my Cas9 plasmid with 50 microliters of sterile water. After this is done, I'm going to take one of my LB agar plates and take my E. coli sample. I'm going to pipette all of the E. coli onto the petri dish and then spread it with my inoculation loop. I'm then going to leave the plate at room temperature for about 48 hours to allow the E. coli to grow. I'm now going to prepare my gene-edited E. coli. First, I'm going to add a microcentrifuge tube onto my tube rack, and I'm going to take a bacterial transformation mix which contains polyethylene glycol and calcium chloride. I'm going to add 100 microliters of this into my microcentrifuge tube. I didn't film this part, but after adding the transformation mix, you should scrape off some of your grown bacteria from the previous plate and add it to the transformation mix. Then you are to mix it until it gives you a cloudy mixture with minimal lumps. The mixture should be cloudy but not open. I then took my tubes of freeze-dried DNA, starting with my Cas9 DNA, and added 10 microliters of this into my microcentrifuge tube. I repeated these steps with the gRNA and my template DNA. After all of the DNA components are added in, I give the bottle a little shake and leave it to incubate on ice for 30 minutes. You can also leave it in the fridge, but you cannot freeze this tube. After my microcentrifuge tube is incubated, I'm going to place it in a cup of 42 degrees Celsius water. You can estimate this temperature by using water that is very warm, but comfortable enough that you can keep your hand in it. I'm going to leave this for about 30 seconds. I then took 2 LB media microcentrifuge tubes and added 1.5 milliliters of room temperature water to each of them. I then shook them to dissolve the lysogeny broth. I'm then going to take my pipette and add 250 microliters of lysogeny broth media to my cell mixture. I'm usually allergic to latex gloves, which is why I didn't wear them for the entirety of this video, but I felt that this was the most important stage where I would actually be editing the E. coli. Once my components are added in, I'm going to leave the mixture at room temperature overnight so that CRISPR can do its thing and replicate the DNA. After waiting overnight, I took my edited E. coli and placed it onto my strep can era plate. I spread it around with an inoculation loop. If my experiment is a success, I will see white or yellow growth after 36 to 48 hours of leaving it at room temperature. I initially thought my experiment didn't work, but here's what it looked like after 4 days. As you can see, there are yellowish colonies of growth. Here's a side-by-side comparison of the non-edited and the edited DNA. This is the edited DNA, and I really hope you enjoyed this video. Thank you so much for watching.